Oh, good. Why don't we go ahead and get started? It's a real pleasure to introduce Jim Wilson, who's going to be giving our grand rounds this morning. By way of background, Jim grew up in Mississippi, went to medical school there, and eventually ended up as a house officer and a fellow at Duke University, where he did a lot of his medical training, and then came up to the Brigham and Women's Hospital. I guess, I don't know if it was the Brigham and Women's Hospital at that time, and did another rheumatology fellowship. So Jim was really focused in the area of inflammation and its intersection with vascular disease and other pathologies. Jim, after a few years as an attending at the Brigham, moved back to Mississippi, where he's been ever since, rising through the ranks as ultimately as professor of medicine. Jim really, about 20 years ago, sort of pivoted and really helped launch the Jackson Heart Study, and really kind of brought the Jackson Heart Study into the modern genomic era, and gets an enormous amount of credit for that. And he's known really throughout the medical community as someone who is really thought deeply about care for underrepresented populations, and also science in those populations, and in bringing that forward. He's been instrumental in leading the TOPMed program at the NHLBI, which is essentially the NHLBI's personalized medicine program. And as I was looking through Jim's CV last evening, I kind of wondered how he pivoted towards genetics, and how, as someone interested in rheumatology, ultimately became very interested in cardiovascular genetics, and really one of the national leaders. And then I noticed that really early on in his work with kind of three of the Illuminatis of inflammatory diseases, K. Frank Austin here, Ralph Snyderman, Doug Fearon, I saw a number of papers in which Jim, you couldn't sequence DNA at that time, but you could chop it up with restriction enzymes and look for different size chunks. And that was kind of the precursor to human genetics. So chance favors the prepared mind, and Jim has a very prepared mind, and I really look forward to hearing him speak today. And I'm sure all of you will enjoy hearing him compare and contrast diseases in whites and in African Americans, and how much more we need to know. So thank you very much, Jim, for giving grand rounds.
Thank you, Rob. I'd like to thank Artie for asking me to speak today, and thank Rob for that very kind introduction. As he said, I'm Jim Wilson, and I'm a professor emeritus from the University of Mississippi School of Medicine. And for the last 20 years, I've worked with the Jackson Heart Study in organizing the study and in recruitment, and particularly in developing the genetic component of the study. And it has been a very good time to be a genetic epidemiologist, which is what I became, because during this period, we really realized that you needed very, very large numbers of study participants to do genetic epidemiology. So we developed a lot of new mechanisms of collaboration and joint data analysis to enable the studies we've done over this period. It's been an exciting time to be a genetic epidemiologist. I have nothing to disclose. So in this talk, I will introduce the Jackson Heart Study, which is where most of my work has been done. I'll talk about some of the distinctive clinical and genetic features of African-derived populations, discuss some of the challenges of genome-wide association studies and efforts to address ethnic disparities in genetic knowledge and eventually clinical care. I will describe a technique called Admixture Mapping that can be used to identify ancestry-specific causal variants, and we'll give some examples of findings with that technique. And finally, I'll discuss some initial findings of proteomics and metabolomics in Jackson that have been done with Rob Gersten's group. And when I retired from Jackson, Rob and I still had a couple of active grants together, which we do now. And so I came up here to work with his group. That has been and is an immense pleasure. He has a large number of gifted young investigators in his group, and it's an exciting place to be. Here is the staff of, this is an early photo of the staff of the Jackson Heart Study. It's a multi-institutional study involving Tougaloo College and Jackson State University, which are two historically black colleges and university, and then the University of Mississippi Medical Center, which is the only academic medical center in Mississippi. It's community driven, and if you look at this photo, you'll see that the Jackson Heart Study staff look like the community that is the target population. In fact, many of the staff are participants in the study, and so it really has been a community-driven study in which we've had a lot of interactions with the community and ongoing contact with participants. There have been 3 exams from 2000 to 2004, 2005 to 2008, and 2009 to 2012. And exam 4 is in progress, though it is somewhat on hold because of coronavirus. Jackson Heart Study, like really all of the other NHLBI studies, has been modeled after the Framingham study, and we collected demographics, medical history, a lot of psychosocial history to explore some of the psychosocial factors that affect the health of African Americans. Collected information on medications, anthropometrics, blood pressure, ECG, extensive biochemical testing, and some very nice imaging, carotid ultrasound, chest and abdominal CT, cardiac MRI, and we prepared genomic DNA, and also in about 1,500 members of the Jackson Heart Study family component, we cryopreserved mononuclear cells for further work. And then over the last 4 years or so, working with Rob, we've done metabolite and protein profiling, and then there's ongoing surveillance and events adjudication. So at the outset, we recruited 5,306 African Americans, which was about 5% of the African American population of Jackson and the surrounding area. Ages 21 to 94 with a median age of 54 at baseline. So at this point, 18 to 20 years out, it is getting to be a study of a fairly geriatric population. 64% are women. There's a nested family study, which includes about up to a third of the overall study, including first, second, and third degree relatives. As I said, we have an average of about 18 years follow-up. And over the last 5 years or so, we've obtained whole genome sequencing, metabolomics, and proteomics in around 3,000 participants. So why do you want to study African Americans? Well, this slide shows U.S. Death rates attributed to heart failure, broken out by age, race, and sex since 2008. And in the first column, you see ages. The next 2 columns are men, with the black men on the left, and the last 2 columns are women with black women on the left. And you can see that particularly in the earlier age groups, for men, there's about a 5-fold excess of deaths from heart failure in this age group. From 45 to 54, there's still a strong difference with more deaths in African Americans, and that persists on through older age groups. The same trends are present in black women. So deaths from heart failure are much more frequent in deaths per 100,000 in black compared to white Americans. Well, what about coronary heart disease? This slide shows the deaths per 100,000 population from coronary heart disease during a 10-year period from 1999 to 2008. You can see once again, black individuals shown by the dark black line are at the top for death rates. And while their rates of death are declining, they remain at the top throughout this period, followed by whites is the next group down, then Hispanics, and then American Indians, and then Asians. So once again, African Americans have drawn the short straw here and are dying of coronary heart disease much more frequently than those in other ethnic groups. So what's the biology behind this? This is from a study by Diane Bild in Circulation in 2005. And they looked in the Meiosis study, which is a multi-ethnic study, where they had coronary CTs in 2,600 white individuals, almost 2,000 African Americans, about 1,500 Hispanic individuals, and then about 800 Chinese individuals. And you can see, compared to the white reference population, in which about 70% of men and 45% of women had detectable coronary calcium with an Agatston score greater than 0, African Americans had significantly less coronary calcium, followed by Hispanic individuals and then Chinese participants. And then if you look at the relative amount in Agatston scores, you see once again African Americans have much less coronary calcium than European Americans, and somewhat less than Hispanic and Chinese Americans. So this is discordant with the findings for coronary heart disease. So another study done in 2011 by Nance et al. Looked at coronary CT angiography in 151 consecutive white patients and 150 consecutive African American patients. And what they found was they confirmed what was seen in MESA for calcified plaque, and that it was much more frequent in white patients. But non-calcified plaque was just the opposite, such that it was much more frequent in black patients. Mixed plaque was about equal. And if you look at the right panel at non-calcified plaque, quantified in milliliters volume, you can see that African Americans had a mean of about 2.5 milliliters of uncalcified plaque, compared to about 1.5 milliliters in the white patients. So not only do African Americans have more plaque in this measure, but it's more likely to be unstable plaque, non-calcified plaque. So to talk about some genetics, this is a cartoon that represents recombination during meiosis. And this is not an accident. Recombination does not happen because of a mistake. It is a regulated process. It occurs about once per chromosome per meiosis, or per generation. And it's directed to specific sites by a protein called PRDM9. PRDM9 recognizes sequence motifs of 12 to 17 nucleotides, and it directs recombination to so-called recombinational hotspots. There are several other implications from this cartoon. If you look at nucleotides on either end of a chromosome, they are quite likely to be separated from each other by recombination over several generations, so that after several generations, knowing the identity of a variant on one end of the chromosome wouldn't tell you anything about the identity of a variant on the other end of the chromosome. On the other hand, if you choose two variants that are side by side on a chromosome, they're quite unlikely to be separated by recombination. And so knowing the identity of one of those allows you to identify the other one with high confidence, to predict the other one with high confidence. And so the two variants at the ends of the chromosome are not predictive of each other, and they are said to be in linkage equilibrium, whereas those that are side by side are highly predictive, highly correlated, and they are said to be in linkage disequilibrium. Finally, there's one other implication of this cartoon that I want to mention, and that is if you have admixture between individuals from African or European ancestral populations or other continental ancestral populations, then over several generations, you don't just have African chromosomes and European chromosomes. Each chromosome becomes a mosaic made up of African and European segments, and you can use that mosaic to analyze certain traits and to look for loci in the genome that are associated with those traits. So based on this and other things, there are several genetic features of African-derived populations that are distinctive. First of all, they're genetically larger than other populations. That is to say, in Africa, recombination was occurring for thousands of generations before just a few people left Africa to populate all the other continents. So the people who left only took with them a small proportion of the genetic diversity that was already present in Jackson. In Africa. I am too Jackson-centric. That was already present in Africa. So that is a bottleneck, and it affects the allele frequencies in other continental populations. Further, because African individuals had been undergoing recombination for a much longer time, the blocks of linkage disequilibrium are much shorter in Africa. As we'll talk about, African, particularly sub-Saharan African populations have been subject to different selective pressures. And then particularly over the last 5 years or so, there's been extensive admixture between African and other continental populations. Finally, African ancestry populations are relatively understudied, as we'll look at in a subsequent slide. So this is a slide that you've probably seen something like in some paper. Genome-wide association studies have been very prominent in genetic epidemiology over the last decade or so. And the way these are done is that usually using a genotyping array, in each individual, there's genotyping for about 1 million different common variants across the genome. And then in a genome-wide association study, you look at a trait, either an outcome like chronic kidney disease or heart disease, or continuous trait like cholesterol, and you test each variant for its association with that trait. So if you're doing a test of 1 million different variants, you've already got a problem, because 50,000 of those will be associated with the trait with p less than 0.05 just randomly. So you have to adjust. You have to set a more stringent threshold for what's really significant. And so just a typical way to do this is to use a Bonferroni correction, in which you divide 0.05 by the million tests that you're doing. And then that yields a p-value of 5 times 10 to the minus 8 for a threshold of significance. So you'll see in this generic GWAS that there are several regions in which variants represented by these single dots are significantly associated with the trait being tested. The other thing you'll see here is that these dots tend to stack up. And that is not because all of them are causal for the trait that you're looking at. It's because somewhere in here, there's a variant that may be causal for that trait. And many of the others are just brought along for association because they're in linkage disequilibrium with that causal variant. And there may be more than one causal variant in this, and that can be determined by first conditioning on the most highly associated variant, and then determining whether there's still a signal left at other variants, and doing that iteratively until you found all the independent signals. So there are several characteristics of GWAS that are challenging. And first is that many of the variants, which are mostly common, that are significantly associated with things of interest, have very small effects. So you need huge sample sizes to detect these small effects. And I've given a couple of examples here. There was a large GWAS of coronary heart disease by Nick Pei et al. In 2015. They had more than 60,000 cases of CHD and more than 120,000 controls. And this brought the total number of CHD loci to 58. So when I say a locus, that's not a single variant that's associated with a trait or an outcome. It is a region. It's a place in the genome, and it spans a certain distance for which there is strong statistical evidence for association with the trait or outcome. And almost all of the lead SNPs, the most highly associated single nucleotide polymorphisms, which we'll refer to as SNPs, have a minor allele frequency of greater than 5%, So the reference allele may have a frequency of 95%. The non-reference allele may have or the minor allele may have a frequency of 5%. And the odds ratios for, in this case, coronary heart disease were quite low, almost all below 1.25. Then this lipids GWAS used 216,000 European Americans, 57,000 African Americans, and 24,000 Hispanic Americans. And I have MVP in red here. This is the Million Veterans Program. And finally, we have genotyping in a large enough number of minority populations that we're going to be able to begin to close the gap in genetic knowledge for diseases that are commonly diagnosed and represented by a diagnosis code in the VA Medical Center hospital record. And so this study looked at LDL cholesterol, HDL cholesterol, total cholesterol, and triglycerides, and identified 562 loci, places in the genome, that were strongly associated with these lipids. And these were largely common lead variants, but in total, they only explained about 10% of overall phenotypic variants. So despite having this huge population where we're only able to identify variants that accounted for a small minority of total variants. So this next slide, if you've heard genetic epidemiologists talk, you will have seen the left side of the slide, and this is from the GWAS catalog maintained by NIH. And each of these little colored gumballs here is a trait or an outcome, and it has been mapped to its place on each of the 23 chromosomes. And so you can see that through a massive international effort, thousands of traits have been mapped to many thousands of loci in the genome. But if you look at the right side of the slide, only 2.4% of the individuals in all these studies were of African ancestry. Only a little over 1% were of Hispanic ancestry, and 78% of the individuals were European ancestry. However, 7% of the associations were from African ancestry individuals, showing that they are punching above their weight. And this may be partially because of increased genetic diversity, but largely it's because most of the low-hanging fruit is still present in African ancestry populations. The same is true in Hispanic ancestry populations. 4.3% of the associations have been found here, whereas fewer associations have been found in European ancestry individuals, proportional to the number of individuals analyzed. So a major project was undertaken in 2014 to address this, and this was really a visionary thing by NHLBI. And so they initiated a project to do whole genome sequencing and other omics, that is transcriptomics, metabolomics, which are being done for TOPMed by the Gersten labs, proteomics, also Gersten lab. And they recruited observational, case control, family-based, and other study designs. I chaired the steering committee for this beginning in 2014. And we have targeted really a total of 145,000 samples from 72 studies so far. And for these, 118,000 samples have already had their whole genome sequence deposited in dbGaP. And one of the goals of TOPMed has been to share these data broadly, both through dbGaP and through a new platform called Biodata Catalyst. If you're going to have this much genetic information on 145,000 people, and you're also going to have many other measures, then it doesn't make sense to mirror all that data in every academic center. Rather, this has been developed as not only a data storage platform, but an analysis platform where approved individuals can go in and use analytic tools to analyze this very large data set. This next slide shows how the ancestral origins of the people in TOPMed are an improvement over what's been seen in the past. 40% are still of European ancestry, but 32% are of African ancestry, 16% Hispanic Latino, 10% Asian, and then 2% other. And this is populations like the Samoan adiposity study of Pacific Islanders. This next slide shows a TOPMed resource that you can use. If you're interested in a particular gene, say, or a protein, say PCSK9 that's involved in lipid metabolism, then you can go to the Bravo server, and you can just Google Bravo server, and you can look at the analysis of that gene. And down in this panel, what you'll see is the coverage for each exon of PCSK9. And you can see that the coverage is very even across the gene. And by coverage, I mean the number of times each nucleotide in that gene has been sequenced in each person. And the average here is about 40 times. So each nucleotide has been sequenced 40 times in each of so far 118,000 people. So this is a tremendous amount of data. And then you can go down to a different part of the site, and you can see that you can see every variant that's been observed in a human being in this population, and you can see the consequence of that variant. So frameshift variants are very disruptive, and so these are all frameshift variants, and these would be expected to reduce the production of or eliminate the production of the protein from one chromosome, and might well have biologic effects. So this next slide shows is a return to the question of chromosomal admixture, and it shows a trio, father, mother, and a child. And in this, African ancestry is shown in yellow, yellow, and then European ancestry is shown in red. And because African and European populations were separated for thousands of generations, allele frequencies for many, many different alleles became highly differentiated. And you can use these to predict at each point in the genome whether there's 0, 1, or 2 chromosomes of either African or European origin. And so if you look in this region, this European segment of chromosome 11 here clearly came from the father because the endpoints are coincident. This one probably came from the mother because of this coincident endpoint. Also this one from the father. But if you look here, this one is coincident at one endpoint for the father, but stops here. So that's a site of recombination. Likewise, this endpoint is coincident here, but here's another site of recombination. So you can you can classify, as I say, in each chromosome with high confidence whether there are 0, 1, or 2 copies of African ancestry at each site in the genome. And this was done with David Reich, Anjali Hinch, and others in Jackson Heart Study and other studies in 2011. So this is a cartoon also from David Reich that's used by everybody who talks about admixture association. And it is a mechanism whereby you can test across the genome for an excess of African or European ancestry in association with a particular trait. So if there's a trait that's more common in African Americans or African ancestry people, you can look across the genome to see if there is a site in the genome where there's excess African ancestry, and that site is very likely to harbor a causal variant for the trait or outcome that you're looking at. So we did this in a disorder or a condition called ethnic neutropenia. This was described more than 50 years ago. It's common in African Americans who are below the usual lower threshold of white blood cell count in about 15% of cases. It's apparently benign, and there were reports of familial occurrence. So working with Mike Knowles and David Reich and others, we assessed white blood cell count and African ancestry in the Health ABC cohort and in Jackson Heart Study. We saw a huge peak on chromosome 1, and additional analyses showed by a number of different additional analyses, we were able to localize this causal variant to the same variant that causes the Duffy blood group antigen. And so if you look at the Duffy variant, for people who are homozygous for the European variant or heterozygous for European variant, you see that the total neutrophil count, which is the count that's most affected, is essentially identical. But if you look at people who are homozygous for the African form of this, the null form of the Duffy antigen, you see that there's a monotonic shift to the left to a lower white blood cell count. So looking at the Duffy variant, which had been thoroughly explored in previous work, it disrupts a GATA1 transcription factor binding site upstream of a gene called the Duffy antigen receptor for chemokines, the DARK gene. And this upstream mutation occurs in this region, in the promoter region, and it prevents expression of the Duffy antigen receptor for chemokines on red blood cells, but not other tissues. The reason this was selected to high frequency in African populations was that it's an obligate receptor for Plasmodium vivax, and having the null, being homozygous for the null allele, is completely protective against vivax malaria. Well, it's also known that this Duffy antigen is a receptor for chemokines like CXCL8 and CCL5 that are involved in neutrophil recruitment. And you'll remember that it's a chemokine receptor that's also a receptor for the HIV, human immunodeficiency virus. So these chemokine receptors have been exploited more than once biologically by disease organisms. The Duffy variant accounts for 27% of the total variants in neutrophil count in African Americans, and it's responsible for the entire difference between African Americans and European Americans in white count. So the next slide was not work done by me or the Jackson Heart Study, but it's so important that I want to mention it. Jeffrey Kopp at the NIH and Linda Cow at Hopkins did admixture mapping in individuals who had focal segmental glomerulosclerosis, and found a huge peak again of admixture association, this time on the 22nd, on chromosome 22. And it was subsequently found by Giulio Genovese, working with Martin Pollack, that this was not MYH9, to which it had been originally attributed, but a protein called APOL1, apolipoprotein L1, or a gene that produces that protein. And an APOL1 protein is a part of the HDL3 particle. So it's involved in, it has a role in cholesterol transport. It attenuates LDL oxidation, but it's also lytic for trypanosomes and perhaps some other parasites. So it has a role in innate immunity in protecting from the organisms that cause African sleeping sickness. However, it's also associated with chronic kidney disease. If you look at focal segmental glomerulosclerosis, there's an odds ratio of about 17 for people who have two African alleles for APOL1, two risk alleles. For arteriosclerosis, which used to be called hypertensive kidney disease, but it's more likely APOL1 related kidney disease with hypertension, there's an odds ratio of about 7. For sickle cell nephropathy, a relative risk of proteinuria of 3.4. Severe lupus nephritis, an odds ratio of 5.4. But look up here at HIV-associated nephropathy. In the United States, there's an odds ratio of 29, but in South Africa, where viral counts are likely to be much higher, it's an odds ratio of 89, suggesting that there is a double hit of APOL1 risk alleles with viral load that's leading to this severe nephropathy. And then this next slide from Jeffrey Kopp's group shows that people who have two APOL1 risk alleles have much faster progression to end-stage kidney disease than people with either 0 or 1 risk allele. So overall, people with 2 APOL1 risk alleles, which is the case in about 1 in 7 African Americans, 20 to 25 of those with 2 risk alleles may eventually develop end-stage kidney disease. So 1 in 4 of these 1 in 14. However, 75 to 80% don't develop end-stage kidney disease. So it's important that we figure out what's different between the people who do and don't get APOL1 related kidney disease despite having the genotype. So far, no interventions have been identified that reduced end-stage kidney disease in this group. So this is a tremendous public health problem in African Americans. Some of you may remember that Cricket Seidman's group led an analysis in Jackson Heart Study in which we assessed the association of APOL1 risk alleles with a combined outcome of myocardial infarction, stroke, or vascular procedures. And we used this combined outcome because we were relatively underpowered, having only about 1,000 people at the time who had been genotyped for the APOL1 variants. And there are 2 variants that lead to disease called G1 and G2, but any combination of these 2 risk variants increases the likelihood of chronic kidney disease. So looking at our combined outcome of cardiovascular disease, we saw that with any combination of 2 risk alleles, there was about a 2 to 4-fold increase in cardiovascular outcomes. This was replicated in the Women's Health Initiative, and subsequently it's been analyzed in the Million Veterans Program using genotyping in about 25,000 African Americans. And this finding of increased cardiovascular disease was confirmed, but it was also found that if you adjusted for incident CKD, then the association with heart disease went away, indicating that these cardiovascular outcomes are mediated through chronic kidney disease. So let's go next to the original molecular disease, sickle cell disease, which all of you know it results from a single nucleotide substitution that results in a change from glutamic acid to valine at the 6th amino acid of the hemoglobin beta chain shown here. Heterozygosity or homozygosity protect against malaria, but in homozygous individuals, deoxygenated hemoglobin tends to polymerize and form these sickle cells. And homozygosity causes anemia, painful crises, and a clinical disease that you're all familiar with. Well, what about heterozygous disease? It's always been considered to be benign, but kidneys have a high oxygen consumption and extraction rate. The renal medulla is highly acidic and hypertonic, and those conditions are ideal to promote hemoglobin S polymerization, even in people who are heterozygous for sickle cell disease. So Rocky Naik from Johns Hopkins contacted us and wanted to see whether there was evidence of increased CKD in people who were heterozygous or who had sickle cell trait. And so in the first 3,000 individuals we analyzed in Jackson, we found indeed that there was a strong signal of increased CKD. So we extended the study through collaboration to include participants from the ARIC study, the Cardia study, Women's Health Initiative, and multi-ethnic study of atherosclerosis or MESA, for a total of about 15,000 individuals. And what Rocky found was for prevalence CKD and prevalent albuminuria, there was a 1.5 to 2-fold increase in association with sickle cell trait, whereas relative risk of incident CKD and rapid glomerular filtration rate decline was similarly increased. And in subsequent studies, Rocky has looked at participants in the very large REGARD study and found that there's also about a 2-fold increased risk of end-stage kidney disease. So let's go back to the issue of coronary heart disease in African Americans. And this is a study by Joshi et al. In the Jackson Heart study and in the Framingham offspring cohort study done in 2016. And Joshi and et al. Compared total cholesterol, HDL cholesterol, LDL cholesterol, et cetera, as you can see on the left. And what they found was total cholesterol was a little lower in African Americans. HDL cholesterol was higher, significantly higher. LDL cholesterol was lower, and non-HDL cholesterol was lower, and triglycerides were substantially lower. So all of those would seem to be a favorable profile with respect to CHD. However, LPA cholesterol was higher in African Americans, as were remnant lipoprotein cholesterol and intermediate density lipoprotein cholesterol. And this finding in LPA cholesterol had been known for some time. This was an earlier study by Markovina et al. In 1996, and they looked at LPA mass, not LPA cholesterol, but LP little a protein mass in about 2,000 African Americans and 2,000 whites. What they saw was that the African Americans had a higher overall amount of LP little a because of a shift to the right in distribution of LPA concentrations. And when Rahul Deo looked at this by admixture mapping in the Jackson Heart study in 2011, what he found was that there was a very strong peak of admixture association associated with increased African ancestry and higher LPA in the Jackson Heart study. And he used the techniques that were current at the time. He did additional fine mapping genotyping. And he found that 21 of the 24 SNPs that were associated with higher LP(a) had higher minor allele frequencies in the African ancestral subpopulation. And he found that common LPA variants explained about 7% of the overall variance in LP(a) and about 73% or more of the contribution of local ancestry to LPA variants. So in this case, LPA, LP(a) is largely controlled by variation at the LPA gene, and subsequent work in TOPMed by Pradeep Natarajan and others has shown that overall about 50% of the variance in LP(a) is associated with a genetic risk score derived strictly at the LPA locus. So this is an interesting finding because Rahul found this in about 4,000 people, totally unlike the studies we talked about of coronary heart disease and lipids, overall lipids. So this is all are largely associated with variation in a single gene. And you'll see that this is the case with metabolites and proteins as we move forward. So this is what we've done in this cohort, thanks to the Robert Gersten group, is aptamer-based proteomics, which use single-stranded DNA molecules. As you know, the sequence of a DNA strand controls its tertiary conformation. And these are about 50 mers. A very large library of them was made by the SomaLogic company, and they were selected for affinity with specific plasma proteins. In our studies, we've used a panel of about 1,310 aptamers. I will use the term proteins for aptamer analytes hereafter. They don't all represent specific discrete proteins, but largely there are 1,310 more or less proteins measured by this panel. And then Rob, for a long time, has been using liquid chromatography and mass spectrometry to characterize small molecules based on their physical features, that is charge or hydrophilicity, and also their mass measured by spectrometry. This is done in Rob's lab at Beth Israel, but also in Clary Klish's lab at the Broad. And they can distinguish, a very powerful technique, they can distinguish about 400 named metabolites and about 4,000 unknown peaks. So I want to talk first about genetic analysis of proteomics. And what we did here with about 2,000 individuals where we had proteomic data, but also whole genome sequencing data from TOPMed for 2,000 Jackson Heart Study participants. This work was done by Akil Pampana, working with Alex Dick and Pradeep Natarajan at the Broad. Akil adjusted the proteins by age, sex, and batch, and then rank inverse normalized. And he tested the association of each of 1,305 proteins with each of about 28 million variants that were limited to those with a minor allele count of at least 5 people showing one copy of the variant. So large number of proteins, large number of variants. Proteome-wide association with each of these proteins was done using a mixed linear model, adjusted for age and sex, and then filtered to p-values of less than 5 times 10 to the minus 8 to make a tractable data set. What Akil found were more than 100,000 variants that were associated with more than 1,000 of the 1,300 proteins at a fairly stringent p-value. A lot of these variants, you'll remember that GWAS that showed that many of the variants stacked up in peaks, and in which there were one or a few causal variants, but a lot of other variants brought along because of linkage disequilibrium. So Akil did a sort of poor man's job of addressing linkage disequilibrium by choosing the most highly associated SNP in a region, and then removing all strongly correlated SNPs that correlate with that highest SNP with an R-square of greater than 0.5 in a 1 million nucleotide window. And this was repeated iteratively for the remaining SNPs in order of decreasing significance. So after clumping in this way, and there are more formal ways of doing this that will identify all of the signals, but after this approach to clumping, Akil retained more than 26,000 variants at 775 loci that were associated, again, with more than 1,000 of the proteins, or about 80% of the proteins analyzed. This is the overall Manhattan plot. What you can see is that these association signals are distributed across essentially all 23 proteins, well, all 23 chromosomes. This little red line down here is a p-value of 5 times 10 to the minus 8. So you can see there are just a ton of findings that would reach what's considered typically genome-wide associated significance for GWAS studies. And some of these associations have p-values less than 10 to the minus 250. So we're looking at 2,000 people here, not 200,000 or nearly a million, as in some GWASs. And yet in these 2,000 people, we're finding very strong associations with proteins. And we're finding the same with metabolites, suggesting that the proteins and metabolites are really quite proximal to the gene, and that we're able to identify genetic effects very powerfully. So there's a great deal of pleiotropy, that is, more than 30 loci are associated with more than 10 proteins. So the APOE locus, the ABO locus are associated with 200 levels of 252 proteins, 130 proteins respectively. So highly pleiotropic effect. And then there are 23 proteins that are associated with more than 100 loci. So there are many, many genetic loci that appear to be influencing the levels of these 23 proteins, showing that their regulation is highly polygenic. And so I want to look at some of the reasons that African Americans may differ from other populations. This work was done by Dan Cruz and in our group, and then Laura Raphael at North Carolina. And they estimated just global ancestry, that is the proportion of African ancestry in 1,500 to 2,000 individuals in the Jackson Heart Study, using ancestry informative markers, that is about 1,500 markers that are highly differentiated between the African and European ancestral populations. And then they analyzed the association of global ancestry with proteins in 4 models. First, just age and sex adjusted, then seriously adjusted for additional measures like BMI, diet quality score, smoking status, estimated glomerular filtration rate, diabetes, lipid levels, and blood pressure. They didn't lose any proteins. There were still strong associations with global African ancestry. Then Dan is particularly interested in environmental effects in minority populations. So they did age and sex plus income and education, which are some of the better surrogates for socioeconomic status in the Jackson-Hart study. This only reduced the number of proteins that are associated with African ancestry to 123. If they did the full adjustment here, plus income and education, still 121 proteins were associated with African ancestry. So clearly, income and education, that is socioeconomic status, affect the associations with some proteins, but other proteins remain strongly associated with African ancestry. Typically, because of the size of the LD blocks being tested, or the ancestry blocks being tested, a p-value of 1.6 times 10 to the minus fifth is used in admixture association. And then testing for 1,305 proteins, a Bonferroni correction leads to a threshold of significance of 10 to the minus 8th. So there were over 1,800 participants with both ancestry informative markers and protein measures. Among these, 66 of the 1,305 proteins showed this very stringent level of significant association with African ancestry. These are some of the, and then they did, instead of just looking at global ancestry, that is proportion of ancestry, African ancestry in each individual, they did actual admixture mapping where they looked across the genome at whether there were 0, 1, or 2 African chromosomes at each locus, and they tested for association with these proteins. And they found a large number of proteins that were associated with areas of increased or decreased African ancestry across the genome. And this includes several different classes of proteins. Here's an adhesion molecule. Here's APOL1, the CKD risk gene. Interleukin-6 receptors, so involved in inflammatory processes. Lipoprotein A1, involved in cholesterol transport. Platelet-derived growth factor. So here's a growth factor that's associated with African ancestry. So you can see from this that just scratching the surface, they're finding that African ancestry is associated with a lot of different biologic processes. Moving on into metabolites, this work was done by Zuzu Chen. It shows association with of metabolites, known metabolites over here with diabetes, unknown metabolites over here. So you can see that there are a lot of known metabolites like uric acid, some amino acids that are associated with insulin diabetes, but then over here, there are all these significantly associated unknowns. So there's a lot of information left on the table here unless we can identify what these are. And Rob has used genetics to do that in one case. DMGV here, dimethyl guanidinovaleric acid, used to be an unknown, but Rob was looking at, in the Framingham Heart Study, Generation 3, there were 470 participants who had liver CT, and correlating metabolites with liver fat, Rob found one unknown metabolite that was strongly correlated, with liver fat. And when he did a GWAS of this metabolite, it had a strong association with a lot of variants that localized to the gene AGXT2. So AGXT2 was known to be an enzyme. One of its substrates is asymmetric dimethyl arginine, and it deaminates that to a compound called dimethyl guanidinovaleric acid. Well, when Rob bought an authentic standard of DMGV. In fact, it exactly co-alluded with this unknown, named here by its mass to charge ratio of 202.1185. Rob did some other sophisticated analyses, fragmenting DMGV and doing a mass spectrometry, and has completely identified this as DMGV. Well, DMGV, now identified, was shown to be associated with diabetes in both the Malmo diabetes cohort, a Scandinavian cohort, and then Jackson Heart study, with the highest quartile of DMGV having a nearly threefold increase in diabetes compared to the referent lowest quartile. Even when adjusting for BMI and glucose at baseline, these findings are minimally attenuated. So DMGV now is associated with diabetes, liver fat, and then Jeremy Robbins assessed the association of DMGV with coronary heart disease in the Jackson Heart study, and found that those in the highest quartile of DMGV actually had significantly higher hazard ratios in the highest quartile, around 2, for coronary heart disease. So DMGV turns out to be extremely interesting metabolite with respect to several human diseases. So in summary, African Americans have distinctive clinical features that can be attributed to both genetic and environmental factors. African ancestry populations have distinctive genetic features related to their deep genetic history, specific selective pressures, and admixture with other continental populations. I've told you about several exciting new resources for whole genome sequence data, multi-omics, and let me say, Rob and I are depositing the proteomics and metabolomics data into dbGaP, and they'll eventually be in the analytic platform of NIH for analysis by the entire scientific community. Places like the Million Veterans Program or the biorepositories at Vanderbilt and Mount Sinai are using data from health records to analyze very large numbers of individuals who've come in contact with hospitals. So there are lots of new resources for exploring biology in African Americans, but other minority populations as well. And finally, admixture mapping indicates that there are many ancestry-specific genetic factors that may contribute to the ethnic differences in metabolites, proteins, and finally for disease. So I'd like to thank a lot of people shown here at the Jackson Heart Study, Harvard Medical School in the Broad, University of North Carolina, and Beth Israel. I hesitate to name people individually in Rob's group because so many people have contributed. And so I would be willing to entertain questions. I think we're a little before the hour. So any questions?
Thank you so much, Jim. I do have one question. Actually, a question from Rob. If you'd like to unmute yourself, Rob, or I can ask it for you over the chat. There is another question in the meantime from Nick Palmieri, one of our fellows, who says, great talk. Can you discuss vitamin D, FGF23, and PTH vis-a-vis racial differences in heart disease?
Very interesting. I really can't. Because you've asked the question, you probably know more about it than I do. Vitamin D has been, or 25-hydroxyvitamin D has been measured in the Jackson Heart study. So far, I'm not aware that there have been striking findings with that, with any of the outcomes in Jackson. However, Dr. Palmieri, if you have read material to the contrary, I'd be delighted to hear your comments on that. I don't know if you can unmute Dr. Palmieri.
I think Rob probably knows a lot more about this than I do too, but I think there's been some associations with these and arrhythmia with atrial fibrillation. FGF23 has been associated with atrial fibrillation, I think in Mesa and Framingham and a couple other things. I guess not so much coronary heart disease, but I wonder just about vitamin D and racial disparities, you know, just.
Yeah, obviously the African Americans are much more likely to be deficient in 25-hydroxyvitamin D, having levels below 30. But beyond that, I have not personally looked carefully at the association either with metabolites, proteins, or outcomes of the Jackson Heart Study. That's something that would be interesting to do, and you might want to talk to Rob and me about that. It would be quite a straightforward analysis. We have the data, and so you know, I'd welcome further discussion of this. Rob had a question.
Yeah, I really had a comment and a question. So the first is, you know, we're all finding these associations. So everybody's clinically oriented in the audience probably saying, so what, right? I mean, we all should say, so what? The first thing is some of those associations, people are making drug targets. So APOL1 is being developed as a drug target. Martin is working with a company, and other companies are working. And then we heard at one of our journal clubs that drugs are being developed for lipoprotein little a. So that's kind of one area. But the area to work on immediately is some of the stuff that you found in chronic kidney disease, right? So we know that people that are heterozygous for a certain disease that we thought had nothing, you know, sickle cell trait, now they have a huge risk for disease. Are there any clinical studies being done in those individuals with some intervention that might not have worked in a heterogeneous white population, but in black population, such, you know, maybe that's where you try your ACE inhibitor, you know, before people develop proteinuria or something. So are there any studies coming out of Jackson, intervention drug studies that are informed by this kind of information?
There are not intervention studies going on in Jackson, and Jackson is not the ideal population to do this for this reason. APOL1 related kidney disease tends to occur pretty early in maybe the fourth decade, people in their 40s to 50s, so that the people who were going to get APOL1 related kidney disease in Jackson probably had it at the time they entered the study, and some of them actually may not have entered the study because they already had severe kidney disease. And
sure,
and,
but just
and
more broadly, any African American population, are there people using that as a marker to treat early using drugs that we already have that may well work in a more focused population that has a super high signal for that disease?
Yeah, I think the best studies of that, which are incomplete, are being done in the Million Veterans Program, where now they have more than 50,000 African Americans who have been densely genotyped on a platform that is intended to provide maximal genetic information in multi-ethnic populations. And so in the Million Veterans Program, they often get people who come into the system early, and they also have very good medications records. So I think the first studies in this area are going to come out of retrospective analyses of drugs that are already being used to treat, for example, hypertension. So yes, things like lisinopril that are ACE inhibitors that are reasonable candidates. So far, nothing has come out, to my knowledge, that actually changes the course of APOL1 related kidney disease.
That's fantastic. That's fantastic, Jim. I don't see any other questions on the chat, so in the interest of time, we'll wrap up here. Thank you for the wonderful talk and the very, very interesting discussion.
Well, it's been a pleasure, and thank you all.
We'll see everyone next week.